Hello all, good morning. How are you guys? So I think you guys can hear me. I just launching the technical code. Okay, am I audible and is the screen is visible? Please click on yes if I'm visible and audible. If you can't hear me, just check your internet connection and also the audio and video devices. If you still think the problem is from our end, please put a message in the WhatsApp group. So some of our associates will help you out. Okay. Great then. Okay. So we'll start the class, guys. It's already 10 o'clock. Okay, let me start recording. Okay. So in the last class, we are actually discussing about biotechnology, right? So how the process happens and all this kind of thing. So before starting the class, I just wanted to inform you that if you guys are watching this particular video from your school, okay, then please keep one of your like class leader or someone near the computer so you can reply me in the chat. Okay, so that will help me to understand like any topics need to be repeated or any topics need to be slowed down in any particular phase or any particular topic you want more explanations. Okay, otherwise it will become more of like in a single set. Okay, I don't want to do that. Okay, so great. Then if you guys are ready, I will start the class. Before starting the class, as usual, yesterday evening I given a homework, right? The question is very simple. I think you guys see this question and solve this. I believe that. Okay, so let us see like what's the correct answer. The question is simple. A specific palindromic sequence which is recognized by echo R1 is. They're asking what kind of palindromic sequence. Yesterday we discussed palindromic sequence is the one which actually run in the same nucleotide sequence in the forward or the backward side, right? From 5' to 3' and 3' to 5', right? So that is called as the palindromic sequence. And they're asking about echo R1. Remember, this question is asked in NEET 2020. That means not last year, before, like 1 year ago. Okay, that particular NEET examination, they asked this question. As I mentioned in the NEET examination, most probably if they ask about palindromic sequence, it's about echo R1 only. Okay, so majorly I'm saying, okay, I'm not saying compulsorily because that is the one which is clearly given in the textbook. If you go for the ICAR or ISER or an ISER examination or KQA examination, they may ask other palindromic sequence also. The list I given in the yesterday only. Notes also sent to your coordinator group. If you haven't written or took a screenshot, just contact your coordinator in the notes. So the complete list will be there. Write it somewhere so you can read it some other time. Okay. So you guys have to remember the particular, okay, echo R1 having a palindromic sequence of GAATTC, right? GAATTC. Now let us guess where is the GAATTC. Okay, the first option is GAATTC. Okay, but let us check the other option also. B option GGAACC. No, this is not the correct answer. D option GGATCC. No, this is not the correct answer. C GAATTC. So now we end up with the two questions. Remember, you may feel that it's easy, right? But it's easy only. But what you have to remember, always the palindromic sequences are decided on based on the movement of polymerase enzyme. As you know that all the polymerase enzyme work in 5' to 3' direction. Okay. So now we have a two option. In one option, GATTC will be from 3' to 5'. Another option, GATTC from 5' to 3'. Okay. Why I mentioned this particular? Because so think about the C option is A and A is B. That means first option you read is this one. Then what you will think, okay, GATTC here is there, then it's the correct answer, you may think, right? That's also happens when you are in a hurry. So remember, whenever you are checking about a polydromic sequence, you have to remember it is from 5' to 3' because polymerase only run in the 5' to 3' direction. So then which is the correct answer? Not C. The correct answer is option A. Right. Option A is the correct answer. 5' GATTC 3'. And other side 5' GATTC 3'. Okay. Understood? Any doubt? If you have any doubt, please let me know in the chat box. I will repeat it. Okay, so what are the things we studied in the last class? Okay, so yesterday we desired to make a Spider-Man, right? We wanted to make a Spider-Man, and in that particular process, what we did, we identify the host and we identify the desired animal, and we identified where the particular DNA where we wanted is, right? So what we did is we did the isolation, right? We did the isolation of desired DNA. Remember, desired DNA means the DNA which have a character we want. Okay. Then what we did, the DNA is very long and we want to cut it at a specific point because we need a particular gene only. We don't want the complete set of DNA, right? That's too long. We don't, there are a lot of like unnecessary genes will be there. So what we did, we use restriction endonuclease, something like ECOR1, BAMH1, something like that. And what we did, we cut it, right? Now what we have now? Now we have a piece of DNA, like several hundred pieces of DNA, or like 10, 20 pieces of DNA. Think about like this, okay? We have all this piece of DNA. Now the issue is like you have to separate the piece of DNA. How do you separate it, right? For the separation of DNA, we have a mechanism from that we will study today. Okay. If you have any questions from the last class, please let me know. Okay, so let me see. The process of the separation of DNA is called electrophoresis. Okay, remember electrolysis is different from the electrophoresis. Okay, don't confuse them. Both are start with the electro only. Okay, electro means as simple as this. It is using the electricity, right? Phoresis means there are some pores are there or something is happening related to pores, right? Electrophoresis. So what we will do here, it's a very simple process. Okay, very simple process. What we do is like we have some already known information. What are those? We know that DNA is negatively charged, right? DNA is negatively charged. That information we already have. Okay, all the DNA, and we also know that DNA can move in the direction of anode or the positive end. Because it have a negative charge, opposite poles normally attract each other, right? A negative charge is attracted towards the anode because it is having the opposite thing, right? So just remember how we process this particular thing. It's a very simple. Okay. First, we need certain area or a media where we can put our DNA, all this like DNA pieces, we got it by the restriction in the nucleus, right? So what we will use, we will make use of a particular chemical called Agarose Gel. Okay, Agarose Gel. Don't think that this is the only gel available. We also have different kinds of gel, something like polyacrylate gel and all. Okay, but Agarose Gel is the one which most frequently used. What we do, as you can see here, we will put a agarose gel like this, okay, in a flat surface, and we will make a small kind of insertion here. These insertions are called well. Okay, these insertions are called well. Okay, well means so there is an insertion is there where you can load your DNA. Loading means what? When we create an insertion and we'll fill this particular what's the table. When you fill the below, there is our agarose gel. Okay, there we made a small holes. Okay, above that we filled with a buffer solution. Okay, we filled with a buffer solution, so there will be less reaction, but the transfer of electrons should, we can put the electron. Then what happens? Here is our DNA pieces. Okay, here is our DNA pieces. We will pour this DNA into this well. Okay, into this well. This process is called loading. Okay, loading of well. What we are loading? We are loading this DNA fragments. We are loading this DNA fragments to this well, which is present in the agarose gel. Once it is filled, we can make it like several wells also, sometime based on like what are we are doing. Okay. Then you put electrode, negative electrode in the initial phase and positive electrode in the last phase. Okay. What we will do, we will start running the electricity. As I mentioned, all the DNA is negatively charged. Why DNA is negatively charged? Can anyone tell me? Why DNA is negatively charged? Why? Yes, Correct, right? DNA is negatively charged because of the phosphate group it have, right? So you know, right? For the DNA or a nucleotide, we need three of. One is the sugar, deoxyribonucleic sugar. Second one is a nitrogenous base. Third one is a phosphate group or H2PO4 minus. Right? So because of the phosphate group, the particular DNA will become negatively charged. Okay. So because it is negatively charged, it will start moving towards the anode. Everything moving towards the anode. But do you think all DNA fragments move in the same way? All the DNA fragments move in the same way? No, that's not possible. Then how it will move? The smaller DNA fragments, okay, they travel in a much higher speed than the larger fragments. Okay. Look, think about something. Agarose gel is there, which have hundreds of like crores of small pores in between. Okay. You put a DNA in this well. Think about one DNA this much small. Then how it will be? All the pores will be enough. Sorry, all the pores, whatever present in the agarose gel, it can pass through easily and it can reach quickly. Think about another DNA is like this much larger. Then what happens? It can't go that much speed, right? It will stuck somewhere in the here. So what happens here? All the DNA fragments start moving towards anode. But the DNA fragments which are smaller will move faster, and DNA fragments which are larger, which normally move slower because the size. Okay. So we will run it for a certain time. Okay. Then what we will do, we will stop this and we will separate this agarose gel. Okay. Now remember, all the DNA what we put in these wells are in between this agarose gel. Don't think that it's still remaining here. It already in between some here. Okay. Can we see in here? Can we see like where is the DNA? Where is our DNA? And no, we can't see it. Okay. Why we can't see it? Because the DNA is not visible to us, to the naked eye. Okay. Then what we will do to identify that, we will add a chemical. What is that? ETBr or ethidium bromide. Okay, ethidium bromide. Okay. This is a kind of, you can see, stain. Okay, so you can just remember like that. Think about like bacteria. Bacteria is not visible, so the crystal violet, so now it's visible. I think you did an experiment with the plants also. You take a section of the leaf, but you can't differentiate xylem from, and also you put a saffron in or some chilacid green or something, saffron in naturally, right? So then you can see this particular structures clearly. Same here, we can't see the DNA, so we put a particular chemical called ethidium bromide. It will go and attach to the DNA. Okay, when you put this particular agarose gel, now in the UV light, what happens? This ethidium bromide will shine. Okay, there's a small chamber will be there. Okay, what we will do is like we will take the agarose gel and we'll keep it under the chamber. Okay, when we close it and we'll turn on the our UV light. Okay, when UV light is turned on, then we can see where is our DNA. Okay, where is our DNA sequences are, where is our DNA fragments are. Okay, so where is this, where is this, and all. What we have to do then, think about this is our desired DNA. We have to cut here. Okay, then we will separate it. Then we will cut and separate the complete gel. Okay, so this is the electrophoresis. Simple. You are taking all the small pieces of DNA, you are feeding it to this particular well, you are putting electrodes. DNA is negatively charged because of the phosphate group, so it moves towards the anode. Okay, after running for some time, you will separate this particular agarose gel plate and you treat it with ethidium bromide. And after treating with ethidium bromide, you check in UV light so you can see where is the chains are. Then you cut that particular gel where your desired DNA is. Okay, this is a very simple. As you can see here, longer bands. So that means longer DNA will be in here, and shorter bands or smaller DNA will be in the tape, because smaller DNA can move or move through this agarose gel more quickly than the longer bands. Okay, understood? So if you understood, I have a question for you. Okay, this question is asked in NEET 2017. Okay, so the question is, DNA fragments are negatively charged, neutral, either positively or negatively charged depending upon their size. positively charged. Okay, I'm giving you 30 seconds of time. As I mentioned, if you are watching from the school, please keep your class leader or someone in front of the computer or the laptop so he can reply in the chat. Okay, whatever your answer may be. So it helped me to understand you guys understood the concept or not. Okay. Awesome. I got a response from Chain Sangli. What about others? Okay. Let me see. What is the correct answer? 30 seconds are over. Okay. They're asking about DNA fragments are. Okay. Whatever the DNA fragment may be. Okay. We'll go for one. Is it positively charged? No. We mentioned it have phosphate group. So it's not positively charged. Either positivity, negativity, charge, depending on their size. Does it matter, guys? This based on size, if it's small positive, large negative, is there anything like that? No, every fragment should be a same. Now, what is that? The charge will be there. So this is also not a correct answer. Is it neutral? No, this is not neutral. Then what's the correct answer? The correct answer is negatively charged. Whatever the size, whatever the thing may be, all the DNA fragments are negatively charged. Jamie Sangley, correct answer. Congratulations. Okay. So shall we move on to the next one? Yeah. So now we have a piece of DNA. Okay, let me tell you. Okay, now I have a piece of DNA which have a desired content. Okay. What shall I do? Shall I go with the experiment? No. The issue is, think about something, you are doing the experiment, you have one piece of desired DNA, you are doing the experiment, something bad happened, okay, something bad happened, and that particular DNA is now destroyed, okay, now the DNA is destroyed. So what you can do? Where you have to start? You have to start the process again from the, where you find the DNA, how we extracted it, all this procedure. Okay, this is the first major thing. And second thing, think about, we are doing the Spider-Man, right? That is the only spider in the world. We got it by hard luck. Okay. And you extracted the DNA also. Now it got spoiled after you separated the DNA. So what you will do? Like, it's a waste of time and like waste of so much resources, and we don't know you get the second chance or not, right? So what we will do, that's why when we got this small DNA, after that we will multiply into several 100,000 million copies, okay? Because if you lost 100 times also same set of DNA, you will have another billion or million with you. Okay, understood. So for that particular process, we will use a mechanism called PCR, or polymerase chain reaction. I think everyone heard about PCR now, right? I don't think there is someone who don't know what the PCR means, right? I know a lot of you don't know what the PCR means, but you heard PCR, right? Where you heard? In Corona, right? RT-PCR you heard, right? So let me explain what is RT-PCR later. First, we will understand PCR first. Okay, PCR is a basic, RT-PCR is a little bit higher level. That okay. So what is PCR, or polymerase chain reaction? Remember, we use this particular polymerase chain reaction for the amplification of DNA, or in a simple term, how can I put this? Means we will use this particular to multiply the DNA into several billion copies. Okay, several million copies. Okay, so how we do it then? Okay, think about like we have a DNA, as you can see here, we have a DNA. It's a 5' to 3' and 3' to 5'. This is a DNA sequence. Okay, this particular DNA sequence I need to be amplified. Before the amplification, or before this, what is the first step? Think about replication procedure. Okay, what happens in the replication procedure in DNA? What is the first step? What is the first enzyme? The first enzyme is helicase, right? What it will do? It will separate the DNA, right? That means what? We also need to separate the DNA because it's a double strand. We need a single strand to make a complementary base, right? What we will do for that? We will put this particular DNA in a machine called PCR machine. Okay, what are the things we will put? We will put some of the things I will write. First one, we will put desired DNA. Okay, that means that need to be multiplied. And what is the second thing you need? Think about replication. Okay, it is getting multiplied. That means it needs a substrate, right? What is the substrate then? The substrate will be dinucleotide, or sorry, deoxyribonucleotide, or how can you nucleotide sequence, or nucleotide bases, right? But what kind of nucleotide bases? Normally, we will use, if you imagine or if you remember the replication properly, okay, we normally have adenosine monophosphate, but we will use adenosine triphosphate as a substances. If you are getting confused, don't think about it. Simply think that. So in the every, what we can say, DNA, if I take the nucleotide, it will look like this. Here is one nitrogenous base and here is one phosphate group. But the substrate which is used in the replication is not this one. The substrate is this one, nitrogen, but here 3-phosphate group. Do you guys know why the substrate is 3-phosphate and the product is single phosphate is joined together? The reason is, if you use this particular one, okay, what is this? Adenosine triphosphate, okay. So when the polymerase chain reaction happens, or when the replication happens, when these nucleotide sequences are combining, then what happens? It will lose 2 phosphates, giving the energy. The particular polymerase need energy to work, okay, it need energy to work. So the polymerase enzyme will utilize this energy and it will activate, okay. So think about simply like a substrate is triphosphate and the product will be monophosphate nucleotide. So that means we need some nucleotide bases here, right? And what's the next step? We need some of the other things to initiate the polymerase reaction. What are those? We need some primers, okay. For the people who don't know what a primer means, just you can think about it. Primer means a short oligonucleotide sequence which initiate chain reactions, or which initiates the replication process. Okay, which initiate the replication process. Okay, understood. So these are small sequence of nucleotide. We say in red primase, which give the primer in the replication. Same. So these are the major things what we need. So we have the content ready, we have the PCR machine ready, we will put all this desired DNA, nucleotide, primer, everything, will keep it, okay. Then we start running. What the PCR machine will do? First, this PCR machine take the temperature or increase the temperature of the inside to a 90 degrees Celsius, okay. This temperature normally varies from 80 to 92 degrees Celsius. Let me keep it in 90 degrees Celsius. When it reaches the 90 degrees Celsius, the first step will happen, that is the denaturation. Denaturation means breaking of double-stranded DNA. Breaking of DS. DS means double stranded. Okay, DS DNA, that means double stranded DNA. As you can see here, 5' to 3' here, 3' to 5' here. What happened? Now it is divided and we have a single strand now. Okay, so this is the first step. That means denaturation. We are given the temperature. Because of the temperature, what's happening? The double strand is breaking to the single strand. Why it is happening? Because in between two strands we can find the hydrogen bonds. Hydrogen bonds are comparatively weaker bonds, so it normally what happens, it will break first. Okay, don't think about it will break in between like this. No, these two strands will be separated. Now I have two strands. What is the second procedure then? I have a primer included. What happens? These primers go and attach to the template strand. This is called as a template, right? Both of the strands are template. What happens? These primers go and attach to the template strand. So as you can see here, we already added the primers. These primers are going and this is getting attached. Okay. You may ask that, sir, when we are in replication, we see that one is a lagging strand, one is a leading strand. Do you think the same thing happens here? Okay, no. what happens in the replication, it's a natural process. There it will go in like that. Okay, here the sequences is very small, and we add two kinds of primer. This is not in the syllabus, there is no need to take attention. Okay, one is a forward primer, another is a backward primer. Okay, that is why the leading and lagging strand formation won't happen in the PCR. Okay, here we will add two different types of the primers. So at a time, what happens, the primer will go and attach to both of the template strands. Okay, then what happens, this annealing will happen not in 90 degree, because attachment won't, the hydrogen bond formation won't happen in the 90 degrees Celsius. So what we will do, this machine will reduce the temperature to 72 degrees Celsius, normally 65 to 72 degrees Celsius. So what happens, the primer will go and attach. Okay, primer will go and attach. Okay, now what is the next step? Polymerase have to come and have to run through the sequence and creating the two strand. That thing is very simple, right? But just check about the temperature. It is 70 degrees Celsius. You know that in the biomolecule chapter of the 11th, we learned that every enzyme work in its own particular temperature, right? One particular temperature, it work efficiently. We call this as a optima, right? We call it as a optima or optimum temperature. Think about the polymerase in our body. It have an optimum temperature of something like 37 to 32 degrees Celsius. Sorry, 27 to 32 degrees Celsius, something like that. Okay. So now imagine, I am putting the polymerase which is extracted from my body in this PCR machine. What happens? Remember, most of these enzymes, or these enzymes are made up of proteins. So protein breakdown will start in right? Protein, what happens in our body, whatever our enzymes is there, it won't become active, it will become inactive, it will get destroyed. Okay, it is not useful. So that is our major hurdle. Then when we are discovering new organism, we come across with a new bacteria. Okay, so the name of the bacteria is, can anyone tell me what is the name of the bacteria? One second. We come across with a bacteria which is in a place of high temperature. Okay, which is in a place of high temperature. Okay, Thermophilus aquaticus. Okay, we got it in a bacteria called Thermophilus aquaticus. Okay, as you can see by its name only, Thermophilus, and phile means what? Okay, phile means which loves. Okay, thermo means temperature, a temperature loving bacteria which is present in the water. That means there are some hot water springs will be there. That means from the ground the hot water will come. Okay, there some bacteria will be living extreme condition. Okay, those bacteria are called a special kind of bacteria group is there. In that one bacteria have a Thermophilus aquaticus, have a polymerase which can work, which can be functional in 70 to 90 degrees Celsius. Okay, we extract the polymerase and we given a name TAQ. That means T represent Thermophilus. Okay, AQ means aquaticus, TAC polymerase. TAC polymerase. Okay, we extracted this and we call it as the Thermophilus aquaticus, or sorry, that's the explanation. Okay, Thermus aquaticus, not Thermophilus aquaticus. Thermus means which love the temperature. Thermus aquaticus. Okay, Thermus means which love the temperature. We extracted this particular polymerase called the TAC polymerase, which can be active in 70 to 90 degrees Celsius. Okay, which can be active in this 70 to 90 degrees Celsius. We put this polymerase. Then what happens? This polymerase will start to run on this, extending the DNA, which is extending the DNA. Okay. So what happens after the end, after when it reaches to the both end, what happens? Now we have a two strands, right? Instead of a one strand, we have a two strand. So what happened here? There is one strand, we put a denaturation, increasing the temperature. Then we decrease the temperature, our primers go and bind each strand, that's called annealing. Okay. Then what happens? The tag polymerase will go and start the polymerase chain reaction. Sorry, we started the extension of the DNA. After the extension of DNA, now we have a 2-strand. 1 strand, 2, we got 2 strand. Then the PCR cycle won't stop here. It will go for the second round. In this, it will go for the second round. Again, it will go for a denaturation annealing extension. After the second round, we got 4. After the third round, we got 8. After the fourth, like 16, then 32, then 64, then 128. Understood what's happening? In every cycle, after revision of every cycle, the number of DNA molecules are doubling. Number of DNA molecules are doubling. Because of that, even in the 20-30 minute cycle, after the 20-30 minute cycle, we will be having millions of DNA strands. Okay, millions of desired DNA strands. Now you guys can do whatever you want. Okay, even though you are wasting 1 or 2 DNA strands, no worry, because we have millions of strands with us. So 1 or 2, if they got destroyed or something happens, no issue. Okay, this is the polymerase chain reaction in a simple. You may ask, sir, we understood this. Then what is RT-PCR, right? Everyone is mentioning RT-PCR. Let me explain with the very small things, guys. Okay, so let me take one screen here. Okay, yeah. So what we seen here in the PCR thing, what we see in PCR, we started with the DNA, right? We started with the DNA and we doubled it, right? We doubled it. Now, with what? With specific primer. Remember, every organism need a specific primer. It's not like the primer which is used for the humans. You can't use it for bacteria, or the primer you use it for the plant. You can't use it on humans. Every organism has specific primer. Okay, so for the extension. Now, think about something. This is the coronavirus. Okay, this is the coronavirus. Okay, coronavirus. What is the genetic material in the coronavirus? The genetic material in the coronavirus is RNA, right? Now, I want to multiply in such a way that I can see that, okay, coronavirus is there or not. But the issue is PCR works on DNA, not on RNA. So what we will do, we will take this RNA and we will put an enzyme called reverse transcriptase. Okay, reverse transcriptase. Okay, I think you know the term. What is the transcription means, right? Transcription means what? Transcription means we are getting RNA from the DNA, right? We are getting RNA from the DNA. What is the meaning of reverse transcription? We are getting DNA from the RNA. Okay. So when we get DNA from the RNA using a technique called reverse transcription using enzyme reverse transcriptase, then this DNA is called cDNA or complementary DNA. Remember what is the meaning of cDNA or complementary DNA? Because the questions are asked from this. Okay, just remember, whenever the DNA, if you obtain the DNA from the RNA using a technique called reverse transcription, it is called cDNA or it's called complementary DNA. Okay, complementary DNA. Okay, now I have a DNA. So what we will do, I will add this in a PCR machine. Our cDNA I will add. Okay, so cDNA of what? cDNA of our COVID. Okay, coronavirus cDNA. Okay. Then I will add coronavirus specific primer. Okay, coronavirus specific primer with a color. Okay, a color coding substances. Okay, either we will use some kind of a stain or the dye. Okay, with a color. Okay, and this color will be activated only when this primer attached to the DNA. Okay, when it attached to DNA, and again nucleotide sequence. We will see two scenarios here. Okay, we have a two scenario here. One, coronavirus is absent. Another one is the coronavirus is present. Okay, coronavirus is present. So what happens here? If the coronavirus is absent, after when you run the PCR, okay, when you run the PCR, okay, here I don't know if cDNA, okay, sample is there. We have a sample here. We don't know coronavirus DNA is there or not. Okay, we have just a sample. When you run the test, if multiplication is not happening, okay, if there is no multiplication or no, what we can say, there will be no amplification of DNA, or the DNA is not getting multiplied. That means what? The DNA, this primer, is not getting attached to. That means it's not a coronavirus, it's some other virus maybe, or nothing is there. Okay. Think about something. If corona is present, then what happens? The DNA will be there, this DNA will attach to primer and it will be multiplied. That means amplification will happen. Amplification means the DNA will get multiplied. Okay, so this process of increasing the doubling of the RNA by converting it to DNA, this is RT-PCR process. Okay, RT-PCR process. You may ask, sir, why we are doubling the coronavirus? We already have thousands of virus among us only, right? We are increasing the number because the virus particle in our body is in a very minute content. Okay, very minute content. What we will do when you put in a PCR, the number of the DNA will be increased, right? Number of DNA will be increased. When the number of DNA number is increased, then you will got to know with this particular color, the primer which is having the color, right? What happens if the number increases? The color also increases. The intensity of color is increases as the number of sequence increases. Based on the color, we will see that, okay, the coronavirus is present or not present. So this process is called RT-PCR, or reverse transcriptase polymerase chain reaction, which is similar to PCR, but the issue is like we will start with the RNA. Okay, it's very simple like this. Okay, I hope you understood. Whenever when you think about next time RT-PCR, okay, think about like how much process we are doing it. Okay, so this is called a golden standards of biotechnology or molecular biology. Golden standards for disease diagnosis. That means we'll definitely find it when we are using a proper primer. Okay, understood. So sometime RT-PCR also called real-time PCR, real-time, because the new PCR machine show that the doubling is happening or not in the screen. Okay, so when the sequence is happening, then only come to know that, okay, doubling is happening or not. That's why it's called real-time. That means in the live we can see something like that. Okay, there is no need to take tension. Remember, RT-PCR not in your syllabus. Don't take tension that sir is teaching this, I have to like remember and all. Okay, just understand the PCR. Now, when question comes, we will see how much you guys know. Okay, the Taq polymerase enzyme is obtained from. Okay, the Taq polymerase enzyme is obtained from. Option is Bacillus subtilis, Pseudomonas putida, Thamus aquaticus, Thiobacillus peroxidans. This question is asked in Need to Learn 16. I got an answer from Sangli. We will wait for the others to answer. Guys who are in the school, as I already mentioned, keep your class leader in front of the laptop or the projector, whatever device you are watching in. Okay, you can reply in the chat, so it will be better for me to understand. Okay. Okay, correct. As the name suggests, T means thermus and AQ means aquaticus. That means correct answer is thermus aquaticus. Sangli students give the correct answer. Awesome. Okay, so remember, there's that much simple it is. Okay, now and remember, along with Taq polymerase, in the nowadays we will also use a different kind of a polymerase, something like Vent, other things also. But for your syllabus, only Taq polymerase is there. Just remember that one only. Okay, correct. Bhimrao also given the correct answer. Okay, shall we move on then? So now we have what? I have a number of desired DNAs. Okay, what is the next thing I have to do? Next thing is like I have to transfer it to a vector or to the vehicle, so I can put it in the host, right? I can put it in the host. These vectors we normally commonly used are the plasmids. Okay, you may ask, what are plasmid? Remember, there's a definition or a proper definition for the plasmid is there. Just remember this or write down. Okay, plasmids are, okay, double standard. Plasmids are double standard. DS means double standard. Okay, circular. That means these are circular. These are self-replicating. These are self-replicating, extra chromosomal, extra chromosomal DNA of bacteria. So this is the definition. You have to remember it, okay? Because if you remember this particular definition, you can solve some problems, okay? Questions. Remember what's the definition of the plasmid? These are double-stranded. As you can see here, it have two strands. These are circular. This will be circle-like. These are self-replicating. That means it have the ability to replicate itself, okay? And these are extra chromosomal. If you think about this is the bacteria, okay, think about this is the bacteria. Here is the chromosomal DNA or genomic DNA. We say these are genomic DNA. That means which normally control the all body structures. Okay, body structure. Our plasmid DNA will be here. Okay, these are plasmid DNA. Okay, these are plasmid DNA. Okay, so these plasmid DNA have the ability for the transformation from one bacteria to another bacteria. So we will consider this particular plasmids as the vectors. Okay, so we can get it from the bacteria only because in eukaryotic you can't find the plasmid, so we can find it only in the bacteria. Okay, so what we will do? So we first check. We will take a natural plasmid. Think about I take a plasmid from the E. coli. This E. coli plasmid do not have any that much character. So what we will do? We will edit it and we will create artificial plasmids. Okay, artificial plasmids. One of the example for the artificial plasmid is, as I seen in the picture, PBR 322. Okay, so what's the name represent then? What's the PBR 322 represent? Remember, PBR represent plasmid of Bolivar and Rodriguez. These are the scientists who created this particular Rodriguez, okay, who discovered this particular, what's that, plasmid, okay, and number 322 means that is the number of the plasmid they created. As you see in the equal R1, the series or the order, 322 is the number of discovery, okay. This is the created by Bolivar and Rodriguez. PBR means plasmid of Bolivar and Rodriguez. Just remember this, okay, don't forget this, okay. Now what we will do or how we will do this, okay. So when we take a DNA, okay, when we take a E. coli DNA, do not have anything in that. We will add a certain kind of genes into this plasmid. Those are called as the cloning vectors. Sorry, those are called as the small, or what we can say, selectable markers. Okay, what are selectable markers then? What are selectable marker? Selectable marker, what we normally use will be a kind of resistance genes. What is resistance gene? Let me explain. Okay. Think about a bacteria. Okay, you have a cold now. Okay, you have a cold. Okay. So what you will do is like you are taking an antibiotic. Okay, you take an antibiotic, but even taking antibiotic also, what happens? Your cold is not get reduced. That means what? Probably you are infected with a bacteria which have a resistance. Okay, which have a resistance to the whatever this one you are using, whatever the antibiotic you are using. Let me tell you, explain you in a better way. Okay. Sorry, my camera stopped working, so I just adjust. Okay. So what happens is like the bacteria, okay, some bacteria will have a resistance to a particular kind of the antibiotic. I will take an example of ampicillin. Okay, so whenever you are taking an antibiotic, just observe what is the name of the antibiotic. Okay, what happens normally if you put the antibiotic, the bacteria should die. But some bacteria could resist the ampicillin. When the ampicillin come inside, they will send it outside, or they will create a protective layer so ampicillin cannot penetrate into the bacteria. So what happens? That particular bacteria will get saved. Okay, the bacteria which gets saved in the presence of antibiotic are called as the antibiotic resistant bacteria. Those resistivity because of this resistance gene which is present in the plasmid. As you can see here, so there are two genes we added. One is the ampicillin and the tetracycline resistance gene. These are called selectable markers. Okay, What are those? One is the ampicillin and another one is the tetracycline. Whenever you are selecting this particular kind of the plasmid, you also need to check about another thing called ORI, or called as origin of replication. Origin of replication. Okay. What is the meaning of origin of replication? Remember, origin of replication is very, very important for a plasmid or any living system, because from here only, okay, from here only the replication starts. Ori means what? Ori means the site of starting of replication, And this Ori also control that how many copies of plasmid needs to be done. As you can see here, genomic DNA one in a bacteria, but the plasmid can be 1, 10, or sometime in a single bacteria you can find 100 plasmid. So the Ori will decide how many times it have to divide, okay, how many times it have to divide, and it also gives a starting point from the replication start. So whenever you are selecting the origin of replication, you have to make sure that you are selecting a proper origin of replication which have a higher multiplication rate. That means higher time it will divide. There is one more site is there called ROP. Okay, ROP means what? Okay, ROP reason, all are small. I write it in this one. Okay, so this ROP will provide. Okay, so or it help in the, not I can't say provide, it will help in the replication. Okay, this ROP region will help in the replication of plasmid, which help in the replication of plasmid. I think that so this is not given in the textbook. Okay, ROP means restriction of plasmid. Okay, sorry, replication of plasmid. Okay, so one simple example I will give you. Okay, 2 years back, in the CBSE final examination, asked that what is ROP. Okay, a lot of students are. I read the biotechnology chapter 2 to 3 times, but I never seen there's an explanation for ROP. This is like out of syllabus. We need to give them like marks, this and okay. But for the people who read the book properly, they come to know that the ROP, what is the function of ROP, is given just below this diagram in your textbook. Open your textbook in biotechnology, just observe PBR 322 diagram is there. Below some explanation is there. There the meaning of ROP is given. That question asked in your final CBSE examination 2 years ago. Okay. That why I'm telling you, because okay, why I'm telling you, because this will give you the importance. Okay, this will tell you the importance of reading every bit of the textbook. Okay, reading the every bit of the textbook. Please read the every bit. Don't ignore that. Okay, I don't need this, I don't need that kind of manner. Okay, so ROP will help in the replication process of the plasmid. What is the next? You can see a lot of these sites, right? What are these sites? These sites are called cloning site. What are cloning site? These cloning sites will have palindromic sequence. This cloning site have the palindromic sequence for different restriction endonuclease. Let me take an example. Check here. Here in the 651 region, what we have? We have a palindromic sequence for SAL1. Here in this region, we have a palindromic sequence for BAMH1. Here in this region, we have a palindromic sequence for E. coli. And here in this region, we have a sequence for the palindromic sequence for the E. coli. These sites where you can find a palindromic sequence for a restriction enzyme is called cloning site. You may ask, so why we call it as a cloning site? What is the advantage of it? Advantage is very simple. When you are taking this vector, okay, as a transport vehicle, what we will do, we will insert our desired DNA, right? Our desired DNA. Where you can insert this desired DNA? You can insert this desired DNA anywhere in these places only, okay? Anywhere in these places where we have a restriction enzyme palindromic sequence. You understood why, right? You understood why. Because think about something, okay, I will take a screen. You have two. You have a plasmid here and you have a desired DNA here. I will cut this desired DNA with eco R1. Okay, eco R1. Then what happens? I have a sequence. I have a sticky end called something like this. Yes, GAATTC. So it will be opposite to that. Okay, G will be C and here will be AA, something like this, right? So think about now, you cut this one, okay, this plasmid with a different kind of the restriction endonuclease, something like SAL H1 or something, okay. Then what happens? You will have a blunt end here, okay, you will have a blunt end here. Both of this will fuse. Can both of this combine or they will fuse? No, right? Not possible. They can't combine because it have a different end. It have a different end. So what you have to do when you are using restriction endonuclease, you have to use same restriction endonuclease for cutting this also and cutting this also. Okay, cutting the desired DNA and also cutting the plasmid. Understood? What I'm saying is, when you use a different set of restriction endonuclease, then the ends are different. So what happens? They won't match, they won't fuse. That's why you have to use the same restriction endonuclease to cut the plasmid and also desired DNA in the initial phase. Okay. So those are the major things. Let me explain. Plasmid, we will use it as extra chromosomal DNA. We use it for the transportation of our desired DNA. It have a 4 major sites. One is the ori, where the replication will start. Second one is ROP, which will give the or help in the replication. Third one is a selectable marker, where we can find the resistance gene, which help us to understand the DNA is there or not. That I will explain in the later stage. Third one is a cloning site, where we can add our desired DNA. These are the 4 measures. Okay. So what we will do then? Okay. Then what we will do is like, we will take this desired DNA. Okay, we will take this desired DNA. Let me explain. Okay, we will cut the plasmid and we will also cut our desired DNA with the same nucleotide, same restriction endonuclease, Then what we will do, we will combine it. Okay, we will combine it. Then what happens? This plasmid will get fused with our desired DNA. It will get fused with our desired DNA. Remember, when we are doing this, you have to remember that you have to select the restriction, or you have to select the cloning site. We have to select a cloning site which is inside a selectable marker, Let me explain why you have to do that. Okay, let me take 2 scenarios. One, I have a plasmid here. Okay, it have a 2 plasmids. Okay, 2 plasmids here. Both of them have a tetracycline resistance. Okay, tetracycline resistance. What I will do, in first DNA, I will add my desired DNA here. Okay, desired DNA in this place, this cloning site. Okay, and in the second one, I have a cloning site here. I add my desired DNA in the cloning site in between the tetracycline resistant. In another thing, I added my desired DNA outside the cloning site. Okay, outside the selectable marker. Then, how do you separate? How do you separate this? Okay, think about, there are a lot of DNAs are there, right? Lot of hundreds of DNAs are there. Then how do you separate this DNA, this whatever DNA from this DNA? This is impossible. You can't see anything, right? You can't see anything. But what happens in this DNA? Just imagine. Because I cut the tetracycline resistant gene and I inserted my desired DNA, now this tetracycline resistant won't work. It won't work. Think about this. There is a DNA which will give a red color flower, It will code for this to here. It will code for some protein. This protein is responsible for red color flower. Okay. Now think about same DNA. In the middle, you added you added some other nonsense DNA. Okay, in middle. So does it give the red color flower now also? Does it? No, it can't give the red color. What happens? Now we are getting a different protein. now we are getting a different protein, right? The protein what we are getting here, will be different, right? So you won't get a red color flower. You may get a white color flower, or you may not get any colors or something else you may get, but you won't get the red color. This particular activity, when you insert a gene, the older gene will lose its activity. This is called as the insertional inactivation. Insertional inactivation. That means what? That simply means when you insert a new DNA, when I insert a new DNA in between a gene, what happens? The older gene will get deactivated. That's called insertional inactivation. Okay. The same thing happens here. What happens when you use your desired DNA in between tetracycline resistant? This particular plasmid lose tetracycline resistance. It will lose the tetracycline resistance. Understood? It will lose the tetracycline resistance. Now I can separate this basically, okay, based on the resistivity. How we separate it, I will explain later. Okay. So shall I move on then? Okay. Now I have this vector, okay, which is added the desired DNA. I want to transfer it to the host, that means to the bacteria. How can I transfer? We have some methods here. Okay, we have some methods. These methods are called gene transfer technique. That means transfer of, how we will transfer of vector. Vector means plasmid, vector or plasmid, to where? To host. How you will transfer, that is the thing. The first step is called particle gun. Here what we will do is like, we will put some of the tungsten or gold beads, okay. Below that we will put our desired DNAs, okay, and below that we will put our cell, okay. So what we will do is like, we will shoot this particular beads, okay, this golden or tungsten beads as like a gun. Then what happens? This bead will pass through this and pass through this. When it is passing through that, it may collect the DNA and it may leave it in the cell. It may collect and it may leave it in the cell. As you know that I'm mentioning may, that means the success rate is very low. Okay. In particle gun, what we will use? We will use either gold or tungsten beads to shoot desired DNA in cell, desired DNA into cell. But the chance of transferring our desired DNA into cell is very low, and you can use it only majorly for the plants, not for the animal cells, because animal cells are fragile comparatively. Now, next one is electroporation. I mentioned it, it's electroporation. Okay, so this electroporation is comparatively a little bit different than that electroporation. Here, what happens is like, we will take one battery plate, we will put our host cells, and we will also put our desired DNA, okay, and we will decrease the temperature, or we will chill it, okay, we will decrease the temperature, and when it is in cold, okay, we will suddenly increase the temperature along with calcium ions, okay, calcium ions, we will sudden increase the temperature, or we will also give sudden electric shock, okay, electric shock. When this happens, what happens? Because of the calcium ions, small pores will be formed on the bacteria fraction of seconds. Okay, small pores will be formed, small holes. Okay, in this chance, the particular DNA may enter our host cell. The desired DNA may enter host cell. As I mentioned, this is one of the good method, but again the chances are comparatively less. Okay. Third one is a microinjection, as we studied in ART, artificial reproductive technique. Here, what we will do, we will take our host cell, and we will directly inject the DNA. inside this. Okay, inside this. This is success rate is comparatively high. Okay, we are directly injecting the DNA into the cell using a micro injection, but the issue is like you need a very skilled labor and very good kind of the what's that laboratory equipment. Okay, these are the major three physical method of gene transfer. These are the three major physical methods of the gene transfer. We will learn about other methods and other 2-3 steps in the next class. Okay, sorry it got a little delayed. If you still haven't understood anything, please let me know. Okay, before coming to next week class, complete your homework and please read the chapter once. So if you have any doubt, I can solve it properly. Okay, thank you for joining. I'm sending the notes in the WhatsApp group. You can ask the coordinators. Okay, if you need, and I'm also sending the PDF of our module to the coordinators. Okay, you can take the Xerox or print out and you can solve the questions. Okay, start reading. Okay, all the best. Yeah, if you have any questions, you can put that in the WhatsApp group. I will reply to that. Okay. Okay, guys, bye bye. We'll meet in the next week. Okay.